Okay, class. So let's start our class. So assalamualaikum and good afternoon. Okay, so we are going to continue our lecture with um, this slide. Okay, so basically during our last lecture we have looked at um, the structure of neurons. Okay, the structure of neurons, and then um, um, we have looked at the cells that support the neurons. So basically, we know that the a neuron, it consists of cell body. Okay, so the cell body, extending from the cell body, it has the dendrites. Okay, the function of the dendrite is to receive electrical signal from the previous neurons. Okay, so the previous neuron that is transmitting impulse, we call it as presynaptic neuron. So the neuron, the first neuron, which is the presynaptic neuron, transmitting impulse to the postsynaptic neuron. So the transmission of impulse will be received by the dendrites. Okay, so the dendrites is extension from the cell body. So and then from the cell body, the electrical signal will be transmitted to the axon. Okay, so before entering the axon, we have the axon helix. So the axon is covered with myelin sheath. The cells of the myelin sheath, we call it as Schwann cell. Okay, so there is gaps. Okay, between the myelin sheath. Sorry, between the Schwann cell of myelin sheath too. So that gap, uh, we call it as gap junction. Okay, so there's gaps lah. We call it as nodes of Ranvier, sorry. Okay, so there's nodes of Ranviers where that section of the neuron is not wrapped by the myelin sheath. So so it is not wrapped by myelin sheath at the nodes of Ranvier. So that is where you are going to find um, voltage gated channel. Okay, voltage gated channel for sodium and also voltage gated channel for potassium. Okay, so basically along the neuron is where the action potential will be generated. Okay, so action potential is when ion move in and out of the axon, causing membrane potential to change. Okay, causing voltage to change. So initially inside a cell it should be negatively charged, but when a cell is stimulated and is generating electrical impulse, the charge inside a cell will change from negative to positive. Okay, so if the change of charge occurs at along the axon, we call it as action potential. It leads to the generation of action potential. So nanti kita akan tengoklah how action potential is generated. Okay, so basically this is the structure of neurons, and then from axon, it enters into synaptic terminal to transmit the electrical impulse. So basically, again, you have to know electrical impulse is due to the movement of ion moving in and out of the neurons, causing membrane potential to change from negative become positive actually. Okay. So okay, so let's look at this slide. So this is where we left off. Okay. Um, okay. So there's neuron can be classified according to, sorry, the change plot. Okay, according to its structure and function. Okay, so functional classification groups the neuron according to the direction in which the nerve impulse travels to the central nervous system. Okay, CNS. Central nervous system, it consists of the brain and also the spinal cord. Okay, the spinal cord. Okay, the spinal cord along your backbone. Okay. So there's the sensory neurons. We call it also as the afferent neurons, and then we have the interneurons, and then we have the motor neurons. Okay, so sensory neurons are the neurons that is it detects changes. Okay, it detects changes. It detects stimulation. Okay, so our body, we have during our the chapter of homeostasis, kan? So our body has to maintain at the right set point of chemical and physical factors lah, kan? So physical factors such as our body temperature, chemical factors such as all that changes of chemical inside our body, such as glucose level, or whatever ion concentration set point, the calcium level, kan. So all that changes, if there's changes occurs to the chemical and physical factors, it will be detected by the sensory neuron in the peripheral nervous system. Okay, so peripheral nervous system detects all those changes detected by the sensory neurons, and then from the sensory neuron, the s- the there will be electrical impulse, okay, that will be transmitted along the neurons until it reaches the interneurons. The interneuron is in the central nervous system, which is the brain and also the spinal cord. The function of the interneuron is to interpret what is happening, what is the corrective mechanism that should be carried out, and then that instruction, the electrical signal, will be transmitted to the motor neuron, so that the motor neuron, which is the efferent neuron, can carry out the corrective mechanism. Okay, so the motor neuron can, for example, be connected to your muscle, causing your muscle to contract. For example, if the stimulus is when you are touching a hot object, okay, so you are not going to maintain your hand on the hot object. You'll be removing your hand from that hot objects. Okay, so that stimulation is through this transmission of impulse. You placing your hand on a hot stove, that sense of hot sensation will be detected by sensory neurons, and then the information, the electrical signal will be sent to interneurons for interpretation of the corrective mechanism, and then the electrical signal will be then sent to the motor neuron for the muscle to contract, you removing your hand from the hot stove, for example. Okay, so sensory neuron and motor neuron are located in the peripheral nervous system. Okay, so motor neuron is also known as the efferent neurons. Okay, so three types of neurons sama je kan. So sensory neuron convey signals from sensory receptor to the nervous system. Okay, so this is it gives you the structure of a sensory neurons. So and then from there, sensory neuron will sense the electrical signal to the interneuron in the central nervous system to interpret, okay, to interpret what is happening, what is the corrective mechanism, can so integrate data and relay signal, and then connect sensory and motor neurons with other nerve cells in the central nervous system. Okay, so that is interneurons. Interneuron is the neuron inside your brains, in the spinal cord. It connects. Dia adalah jambatan lah kan. So from sensory neuron to the interneurons of the central nervous system, and then send the impulse to the motor neurons in the peripheral nervous system. So motor neurons convey signal from central nervous system to the effector. Effector can be your muscle. Your muscle will be contracting, for example, or the motor neurons will be connected to glands, endocrine glands, causing glands to secrete hormones, produce and secrete hormones. Kan? So, or the motor neurons will be connected to another neurons until it reaches the target tissues. Okay. So if you look at this diagram here, it shows you the transmission of impulse along the motor neuron, interneuron, and the motor neurons. Okay. Okay, so if we look at this diagram here, so this is where apa dia auto ni, okay, so this is where you can see a reflection kan, when you hit your knee kan, at the kneecap, and the bawah kneecap tu ada tendon kat sini kan. So that when you are placing pressure in this area, you knock on this area kan, to see the reflex. So that is detected by the sensory neuron. Okay, so that's it detects changes. In this case, it's physical change. Yeah, so physical factors. So it causes a sensory neuron to receive the information, send the information in the form of electrical signal, and then through the sensory neurons, and then reaches the interneurons in the central nervous system for interpretation, integration. Okay, what is happening? What is the corrective mechanism that should be carried out? And so that interpretation of information will be sent through electrical impulse to the motor neuron, okay, to the motor neuron in the central nervous, in the peripheral nervous system. So this neuron, the motor neuron, is attached or connected to your muscle. Okay, in this case, it's quadricep muscle, quadricep muscles, and causes the muscle to contract. So your reflex with your your uh, your your leg will be kicking lah, macam tu kan, due to this reflex. Okay. Sama, okay. So the same explanation. So the sensory neuron, sensory receptor at the end of the peripheral nervous system, okay, peripheral nerves of the peripheral nervous system ni, PNS, detects the body internal and external environment, detects any physical or internal change, okay. So external or internal change, chemical or physical factors. Okay, so this receptor also detect changes that occurs. For example, when you touch a hot object, a sensory receptor picks up that information of pain. Okay, so it detects the stimulus. Okay, the change, physical or chemical factor, internal or external environment. So all sensory information is picked up in the peripheral nervous system by the sensory neurons and sent to the interneurons in the central nervous system. Okay, so and then what happened next is the integration. So that information, that electrical signal will be sent to the uh, to the to the brain or the spinal cord. Okay, so this organ receives sensory information and makes decision. So the interneuron interpret what is happening, what is the corrective mechanism that should be carried out. So and then, for example, if you feel pain, your brain might decide you need to move away from the painful stimulus. Okay, so apa-apa stimulus, apa ketuk dekat lutut ke ataupun usik tempat yang panas, whatever stimulus that cause this the electrical signal to be transmitted. And then from the interneurons, the electrical signal will be sent to the motor neuron in the peripheral nervous system. Okay, the motor neuron, the effector. Okay, so once the central nervous system makes decision, the brain is interpreting. Okay, it makes decision. So it then carries out the motor function. Okay, so during motor function, neurons in the central nervous system carry an impulse along a peripheral nerve to either the muscle or a gland, the effector. So if the neuron is connected to the muscle, it causes the muscle to contract. Okay, if the the motor neuron is connected to a gland such as an endocrine gland, it causes the endocrine gland to secrete hormones. Okay, so let's say, for example, um, connected to your brain, there's the hypothalamus. Kan? So the hypothalamus, it consists of neuron cells. Kan? So the neuron cells in the hypothalamus function to secrete to produce and secrete hormones such as oxytocin. So when the neuron is stimulated, okay, so it triggers the posterior pituitary to release the oxytocin into your blood to reach your uterus, to cause uterus to contract during childbirth. Macam tu kan. So the gland, such as, for example, it can be the hypothalamus causing the posterior pituitary gland to secrete oxytocin. Okay, oxytocin. So that is one of the example. Okay, so the effector is the uterine muscle lah. Uterine muscle contract during childbirth to push the baby out, kan, right? So that is another example. Okay, so we have sensory neurons, interneurons, and motor neurons. Motor neurons connected to muscle or a gland. Muscle contract, gland secrete hormones, for example. Okay. So next, okay. So basically, the function of a neuron, we know that it is to transmit electrical impulse. Okay, The function, I repeat, the function of neuron is to generate the electrical impulse, transmit the electrical impulse. What is impulse actually? It is electrical signal. Okay, usually, okay, for example, saya tambah slide. Okay, I add a few slides here. Okay, okay, usually, okay, normally your body cells, any cells, okay, any cells, it inside cells should be negatively charged. Outside is positively charged. Okay, so same goes to a neuron. So this is a neuron. Okay, the apa nama, the axon, and then we have the axon terminal. Okay, so the neuron is covered by myelin sheath. Okay, we have here is the nodes of Ranvier. Alright. Nombor pula dia transmit gambar apa? Saya dah sketch, dia lambat transmit. Lambat appear. Sekejap. Tu dah ada. Okay. Daging. Okay. We have here is the neuron. Okay, so the neuron, it has this the cell body with dendrite extending. It has the axon, which is covered by myelin sheath. And then we have the nodes of Ranvier, which is the gap between the myelin sheath, right? So, when a neuron is not stimulated, okay, we call this the neuron is in the resting state. Resting state. It is not transmitting or generating impulse. Okay. So for this neuron that is not transmitting or generating impulse, inside the cell it is negatively charged, especially at the axon area. Okay. So when the neuron is resting, inside cell is negatively charged, outside is positive. Okay, outside is positive. So if a neuron is stimulated and it is going to generate an electrical impulse, so it has to change these charges inside and outside of the cell. Okay, so the charge should change from positive, it has to become, sorry, from negative, it has to change to positive. Okay, so outside it changes from positive, it becomes negative. So so this is when the neuron is stimulated and generating electrical impulse. Change from negative, it becomes positive. So what causes the membrane potential to change from negative to positive? It is due to the movement of ions. Okay. So initially the cell is negatively charged. Sodium enters, changing the potential to become positive. Membrane potential to become positive. Kita pause kejap kelas. Saya kelas saya punya laptop ni dia disconnected sekejap. Okay, kita start balik semula dengan explanation tadi ya. Badan kita ni terdiri daripada banyak sel kan. Okay, selalunya sel-sel dalam badan kita ni fungsi dia adalah sel kat tangan ke mana-mana sel lah. Fungsi dia bukan untuk hasilkan electrical signal. Okay, special untuk neuron, dia boleh hasilkan electrical signal. Okay, sel-sel dalam badan kita ni termasuk neuron, asalnya dalam dia negatif. Dia bersifat negatif. It is negatively charged. Okay, so dalam negatif, luar positif. Okay, luar positif. So, okay, luar positif. So, untuk neuron, kenapa neuron ni boleh menghasilkan action potential? Sebab neuron ni semasa dia nak transmit and generate the electrical impulse tu, charge dalam sel, dalam neuron ni kena bertukar dari negatif ke positif. Okay? Dari negatif ke positif. Disebab apa yang menyebabkan charge dalam bertukar daripada negatif jadi positif disebabkan oleh pergerakan ions. Okay? Ion-ion yang terlibat selalunya adalah sodium ion and also potassium ion. Okay? Dan juga adalah chloride juga, negatively charged ion. Tapi kita nak fokus kepada dua ni, sodium and potassium ion. Okay. Kita kat sini, saya draw kat sini ada dua jenis neuron. Kita ada presynaptic neuron, kita ada postsynaptic neuron. Presynaptic neuron is the neuron that is going to transmit the electrical impulse, and the post is the one that is going to receive the electrical impulse. Okay, the electrical signal. Okay, so let's say lah for example, this neuron is transmitting the impulse. Impulse tu, electrical signal tu disebabkan oleh apa? Pergerakan ion kan? Pergerakan ion. So this is the region we call it as synaptic terminal. Okay, synaptic terminal. So this synaptic terminal, okay, dia ada apa? Dia ada vesicle containing neurotransmitter. Okay, neurotransmitter. So the neurotransmitter from the synaptic terminal of presynaptic neuron will be released into the this synaptic cleft. Okay, into the synaptic cleft. This neurotransmitter that is released into the synaptic cleft dia adalah chemical molecule. Okay, it is a chemical molecule. So we call it as ligand. Okay, ligand chemical molecule in this case is the neurotransmitter released into the synaptic terminal, into the synaptic cleft. Okay, so this neurotransmitter, when it is released into the synaptic cleft tu, what is the function? It binds to receptor located on the dendrite of post. Dekat dendrite of postsynaptic neuron, it has an ion channel, okay, an ion channel which we call it as ligand gated. Okay, ligand gated channel. Okay, so this ligand gated channel, kalau kita close up draw dia kan, kalau kita close up draw, dia ada receptor binding site. Okay, so the function of the ligand gated, it binds to the chemical molecule which is the neurotransmitter, which is the ligand, the chemical molecule. So neurotransmitter bind to the receptor, causing the channel to open. Dia adalah channel. Okay, dia adalah channel. So the binding causes the channel to open. So once the channel opens, okay, then only sodium can enter into the postsynaptic neuron. Okay. So at the dendrite region here, initially it is negatively charged. When sodium enters, it changes the negative charge to become positive. Okay, so that's why the neuron, the postsynaptic neuron, is capable of receiving the impulse, the electrical impulse. Pergerakan aliran ion tu masuk dalam dendrite. Okay, dalam dendrite. Okay, so along the dendrite, due to the change of charge, okay, it generates membrane potential. Okay, it generates membrane potential. So in this region here, the change due to the movement of sodium into the cell akan generate potential. We call it as graded potential. Okay, kat sini kita namakan dia sebagai graded potential. Okay, graded potential. So graded potential can only occur along the dendrite, along the cell body until it reaches the axon helix. Okay, so let's say sodium ion enters, it causes the membrane potential to change and causes the generation of graded potential. Okay, so graded potential occurs along the dendrite, along the cell body until it reaches the axon helix. Okay, so this graded potential, if the value is high enough, nanti kita akan tengok dia akan ada bacaan value voltage dia tu. If the voltage reaches certain value, okay, along the axon, it can lead to the generation of action potential. Okay, So action potential that occurs along the axon is due to the opening of another ion channel. So ion channel that is located along the axon, we call this as voltage gated ion channel. Okay, voltage gated ion channel. So voltage gated ion channel located along the nodes of Ranvier of axon ada dua, which is sodium voltage gated ion channel, and another one is potassium voltage gated ion channel. Okay, so when, for example, if the voltage gated sodium opens, it causes sodium to enters, changing the membrane potential from negative it become positive. Sebab sodium masuk. Sebab apa? Voltage gated channel opens. Okay. So if enough sodium enters, it causes a certain voltage to be reached and causes the generation of full action potential. Okay, full action potential. So action potential occurs along the axon. Ingat radiate potential occurs along the dendrite cell body. Along the axon, it triggers action potential to occur. Okay, so membrane it changes from negative, it becomes positive. Okay, so another ion that involved in the movement is the movement of potassium. Ingat kalau sodium it enters, potassium it diffuse out. The movement of these ions semua ni adalah melalui facilitated diffusion. Tak kisahlah dia guna ligand gated ke, dia guna voltage gated ke, the process is facilitated diffusion. The movement of ion is down concentration gradient. Okay, so let's look at this diagram. Okay, so what causes impulse to be transmitted? Transmission of impulse. We know impulse is electrical signal. Okay, electrical signal is due to the movement of ion moving in and out of the cell. Okay, in this case is the neuron lah. We are focusing on neurons. Okay, so we know that the process of facilitated diffusion, it involves either carrier protein or channel protein. Okay, so carrier protein, it can in this case it involves the facilitated diffusion lah. Another function of carrier protein, it involves in active transport. For neuron it does not involve any carrier. Okay, any carrier. So carrier tu untuk contohnya untuk active transport sodium potassium pump contohnya kan, ataupun untuk molekul yang lebih besar such as glucose, amino acids. So we are focusing on transmission of impulse. It does not involve any carrier protein, but it involves several different ion channel. Okay, so this ion channel can be non-gated, can be gated. Okay, so non-gated, it means that the channel is always open. It is always open. It is always open, and because it is always open, we call it as leak ion channel. Why is the term leak ion channel? Kalau contohnya kita ada satu botol air, kita isikan dengan air. Kenapa botol air tu bocor? Sebab air daripada kawasan air yang banyak, dia akan pergi kat kawasan air yang lebih sikit lah kan, leak contohnya. So same goes to the non-gated. Okay, so let's say for example we have this is a membrane. Okay, we have the non-gated ion channel, the leak ion channel. Okay, so outside, so this is the interstitial fluid outside, the extracellular fluid side. Kat luar ni, kat dalam ni kita ada the cytoplasmic side. Okay, so let's say for example, outside the concentration of sodium is high. Outside cell, the concentration of sodium ion is high. Okay, so because it is facilitated diffusion, it causes the sodium ion to enter into the neuron. Okay, to enter into the axon of the neuron. Macam itulah. Okay, the axon of neuron. So sama juga for potassium outside is low, but inside the concentration of potassium is high. Okay, so for this case, in this case, for potassium from inside from high concentration, it diffuse outside. So the movement is always down concentration gradient using the ion link channel. Okay, it is non-gated. It causes ion to diffuse down concentration gradient. Okay, down concentration gradient. But for the gated one, okay, for the gated one, we have three types of gated. Okay, we have ligand gated channel, we have voltage gated channel, we have mechanically gated channel. Okay, so ligand gated channel, take note that it is located at the dendrite of neuron. Okay, ligand is referring to chemical molecule. Okay, so ligand gated channel, as you can see from this diagram, it has binding site. Okay, binding site to which is in this case it is to bind to neurotransmitter. Okay, so let's say imagine kita ada apa membrane of dendrites. Okay, dendrites. So we can find here the ligand gated channel. So when neurotransmitter bind, it causes the channel to open. Sodium enters into the cell. Okay, sodium enters into the cell, changing the membrane potential from negative, it becomes positive. So along the dendrite, there will be generation of graded potential. Okay, so open when appropriate chemical bind to the receptor. Chemical in this case, for example, is neurotransmitter. Neurotransmitter binds, it causes the channel to open, sodium enters. But if the neurotransmitter is removed, the ligand gated channel closed, so the sodium cannot enter into the neuron. Okay, so itulah, so that's why we call it as ligand gated. Neurotransmitter bind, channel open, sodium can enter, for example. Okay, so and then we have another channel which is voltage gated channel. Okay, so voltage gated channel is located along the axon at the nodes of Ranvier. Okay, so it says here open and close in response to changes in membrane potential. Okay, so along the axon, this is the area that is covered by myelin sheath. Kat sini ada dia punya nodes of Ranvier. Kat sini, this is where you will find voltage gated channel. Okay, voltage gated channel. If, for example, there's stimulation, the neuron is stimulated, inside cell it is negatively charged. If the neuron is stimulated, it causes the voltage gated channel to open. Okay, voltage gated channel to open. If this is voltage gated sodium channel, sodium enters. Sodium enters, it causes the membrane potential to change from negative become positive. Okay, from negative become positive. Vice versa, kalau ini adalah potassium, when potassium channel open, it causes potassium to diffuse out. Yang ini kita akan tengok nanti. Sifat sodium, sodium ion akan sentiasa nak masuk dalam neuron, tapi potassium sifat dia sentiasa nak keluar. And the movement of sodium and potassium in and out of the cell is down concentration gradient. It is facilitated diffusion. Okay, so and then we have another one which is mechanically gated channel. Okay, so open in response to physical deformation of the receptor. Physical deformation, for example, kalau you have your hand, your skin kan, so you are pinching your skin, you that is the stimulus, so it opens the mechanically gated channel. It is due to pressure. Okay, physical deformation of the receptor, it causes the channel to open. Alright, disconnected lagi kejap ya. Saya saya nak extend sikit masa lecture kita sebab tadi kita start lambat. Maybe pukul 5 ke 5.10 ke ya. Oh, saya kena tukar lah kejap. Connection internet dekat laptop tak berapa nak elok. I have to use my desktop. Okay. Okay, so let's move on to this slide here. Okay, so we know, we know a neuron is capable of generating electrical impulse. It is due to the movement of ion in and out of the cell. Okay, so now we want to look at all these terms here. Okay, so it says here membrane potential. So what is membrane potential? Membrane potential is when there is difference in charge between inside and outside of the cell. So in this case, normally for a cell, inside cell it is negatively charged. Outside it is positively charged. Take note here, kat sini I'll draw kat sini dalam gambar rajah ni. Okay. This is the extracellular fluid, the interstitial fluid side yang warna biru tu kan. So you can see there's a lot of sodium ion outside. So secara normalnya kat luar sel ni concentration of sodium is always high. Okay. Concentration of potassium, nampak ada satu je kan. Potassium concentration is low. Compared to the inside of the cell in the cytoplasm, you see that the concentration of sodium is low. The concentration of potassium is high. So pergerakan ion-ion ni keluar masuk dalam sel ni is through facilitated diffusion. So maksudnya sifat sodium dia sentiasa nak masuk dalam neuron ni. Sifat potassium dia sentiasa nak keluar. Okay, nak keluar daripada sel, sama ada dia guna voltage gated ataupun ligand gated. Okay, so due to the movement of ions, it causes this the formation of membrane potential. So this membrane potential, we can measure the value which is in terms of voltage. Kan? So voltage is the measure of potential energy generated by separated electrical charges. It is always measured between 2 points. 2 points which is outside of the cell in the interstitial fluid side, extracellular fluid side, interstitial fluid lah, and inside cell which is in the cytoplasmic side. 2 points. Dalam negatif, luar positif. Okay. So plasma membrane of neuron has unequal distribution of electrical charges between two sides of the membrane. So this create electrical gradient. Okay, value yang kita measure electrical membrane potential tu we call it as voltage value tu kan. So because it is cells, so the unit is millivolt. Okay, millivolt. Okay. So membrane potential, this electrical gradient is called as potential difference that exists in every cell's plasma membrane. So the charge difference or voltage across plasma membrane we call it as membrane potential. Perbezaan charge luar dengan dalam ni adalah membrane potential. Kita measure dia sebagai voltage. Okay, voltage. Measurement is voltage. So because it is cells, the unit is millivolt. Tak sampailah volt kan, so millivolt. Alright. So because inside cell is negatively charged, this is at resting potential of a neuron when neuron is not stimulating. So at the resting state of a neuron, the reading at resting state is negative 70 millivolt. Okay. But if the neuron is stimulated to to generate electrical impulse, nanti dia akan generate action potential along the axon ni. The reading will change from negative 70 becomes positive 30 millivolt at the peak of the action potential. Peak of action potential daripada negatif dalam resting state tu dia akan jadi positive 30. Why? Because voltage gated sodium channel opens, sodium masuk. Macam tu. There's a lot of sodium that enters into the axon causing generation of action potential. Okay. Okay, measurement dia adalah dalam voltage. Positive 30 millivolts at the peak of an action potential. Okay. So next, okay, sebelum kita nak fahamkan macam mana action potential tu boleh terhasil along axon, we need to know this resting potential here. Okay. So resting potential. Sekejap ya kelas. Hello. Tengah lecture. Sekejap. Nanti buka Okay. Alright, so okay, resting potential. So resting membrane potential difference in a resting neuron. Neuron that is not stimulated. It is not generating any impulse. It is not stimulated. Okay. So neuron that is not transmitting any signal. So the reading of the resting potential should be negative 70 millivolts. Okay. So there are three factors that contribute to the maintenance of the resting potential at negative 70 millivolt. You always have to put the negative symbol. Okay. Negatif kalau awak tinggal, dia jadi salah jawapan. Okay. So negative 70 millivolt at the resting potential is contributed by three factors. Okay. Three factors. So three factors contribute to the unequal distribution of ions between the interior of neuron and the extracellular fluid. Okay, so the first one is the presence of sodium potassium pump along the axon. Okay, along the axon of a neuron, there will be sodium potassium pump. Okay, sodium potassium pump, we know it involves active transport. Okay, active transport. So it says here, sodium potassium pump actively pumps into potassium and pumps out 3 sodium ions. So this maintain high concentration of potassium and low sodium inside cell, whereas yang ni saya kena draw lah, dia macam panjang sikit kan, saya draw sekejap keep, dah tak ada fikir lah mana nak keep. Okay, saya draw kat sini. Okay. So imagine ini adalah axon. Okay. This is the axon of a neuron. Okay. Axon of a neuron. Kat sini kita ada the myelin sheath. And then the gap is the nodes of Ranvier. Okay. Dekat nodes of Ranvier, there will be several different channels. Okay. Itu tak apa. Okay. Initially, kita nak concentrate pada resting potential. Okay, resting potential. Resting potential inside cell, it has to be negatively charged. It has to be negatively charged compared to the outside, positively charged. Okay. So the first factors that contributes to the negative charge inside cell is the sodium, potassium pump. So you look at the pump, it involves what? Active transport. Okay, active transport. So saya nak you all ingat dan fahamkan perbezaan concentration of ions ni. Dekat luar, in the extracellular fluid side, the concentration of sodium ion is always high. Okay, inside cell, the concentration of sodium ion is always low. compared to the concentration of potassium ion outside is always low, inside potassium ion is always high. Take note of this. Ingatkan dan fahamkan. Okay. So when this happens, okay, what is the role of sodium potassium pump? Okay. So here for example, we have the sodium potassium pump. Saya draw warna hijau lah. Okay. The sodium potassium pump. Sodium potassium pump here. Okay. The sodium potassium pump, it involves in active transport. It means that the ion has to move against concentration gradient. Okay, against concentration gradient. In this case, kalau against, kita tengok sodium. Sodium maksudnya dia bergerak daripada dalam, kan, daripada dalam ni, nampak tak dalam axon ni? Dalam cytoplasm ni, sodium ion is low. Okay, sodium ion is low, outside sodium ion is high. It is active transport. It means that the sodium ion move from low to high. Okay, move from low concentration to high concentration. So sodium ions keluar. Okay, sodium ions move out, is pumped out from the cytoplasm to the extracellular fluid site. Berapa banyak? 3. Okay, 3. For the potassium, you can see that, okay, outside potassium concentration is low. Inside, potassium concentration is high. So the movement will be from outside into the axon, into the cytoplasm. So potassium move into the cytoplasm, into axon. Okay, potassium masuk. Berapa banyak yang masuk? 2. Okay, 2. So how can this process contribute to the negative charge inside the axon? Tengok. Berapa banyak positive charge yang keluar? 3 kan? 3 sodium yang keluar berbanding dengan 2 potassium yang masuk. It means that there will be more positive ions going out and less positive ions that enters into the cell. 3 keluar, 2 masuk. 3 keluar, 2 masuk. 3 positive charge move out against 2 positive charge that move in. So this makes sure that inside the axon is maintained negatively charged. Okay, that is the first point. And then the second point that maintains the cell to be negatively charged is the presence of leak ion channel. Okay, leak ion channel, remember that it is always it is always open. Okay, it is always open. So okay, it is always open. So the second factors that maintains the negative charge, second factors is the leak ion channel. That is, it is always open. So again, we draw the structure of the axon. Okay, so we have here the nodes of Ranvier. The part that is covered is the myelin sheath, and the part that is not covered is the nodes of Ranvier. So the leak, again, you have to take note of the difference in the concentration of sodium tadi. Sodium kita tahu kat luar tinggi kan, dekat dalam rendah. Okay, for the potassium luar rendah, dalam tinggi. Okay, leak ion channel terlibat dalam facilitated diffusion. So the movement of this ion should be down concentration gradient from high to low. Okay, dekat dalam membrane of axon ni kan, there will be the leak ion channel. Saya guna yang warna biru lah untuk potassium. Okay, potassium leak ion channel kan, the potassium leak ion channel will be more, there will be more compared to the sodium lead ion channel. Okay, more. So in this case saya draw 3 kan, 3 untuk potassium berbanding dengan 2 untuk, sorry, sodium saya draw 1 je. 1. Ni contoh lah, contoh je. So there will be more potassium lead ion channel compared to sodium lead ion channel. So when there is more potassium lead ion channel, pergerakan down concentration gradient kan. So there will be more sodium that diffuse out. More sodium, more potassium that diffuse out. Okay, more potassium diffuse out compared to sodium that diffuse into the cell. Okay. Nampak tak? Okay. So when more potassium diffuse out of the cell, there will be more positive charge going out. Okay, going out. So this causes the inside of the cell to be maintained negatively charged. Okay. So ingat untuk lead ion channel, there will be more lead ion channel compared to the sodium lead ion channel. Okay, more potassium compared to the sodium. Kan macam tu. Okay. Pergerakan is always down concentration gradient. The next factor that causes the maintenance of resting potential at negative 70 millivolt tu, okay, is the presence of fixed anions. Okay, again, this is the membrane of the axon. Okay, we have here the myelin sheath covering the axon. Okay, so what are fixed anion? Fixed anion are large macromolecules inside cells that are negatively charged. Okay, large macromolecules that are negatively charged. This macromolecule can be DNA, it can be carbohydrate, can? What is this? Okay, and then it can be proteins. Okay, so this large macromolecule that are negatively charged inside the axon contributes to the axon to be negatively charged. Okay, so this fixed anion is maintained within the axon. It cannot diffuse out due to its large size. So it is inside the cell, inside the axon, contributing to the negative charge of the axon. So there are 3 factors. Okay, so the sodium potassium, the lead ion channel, and the presence of fixed anion. So all these factors you should be able to explain how these 3 factors contributes to the negative charge of axon during potential. Okay, during resting potential. Okay, so kalau kita tengok balik yang slide ni kan, so kita ada pump, kita ada lead ion channel more numerous for potassium, okay, more numerous for potassium, and then compared to the sodium, and then we have the presence of fixed anions. Okay. Okay, so let's look at this video, and then we finish off our lecture. Okay. Okay. So kita stop sampai situ dulu lah kot kan. So there's a lot of input. Taklah banyak sangat input. Semua proses tu you all kena fahamkan dulu kan. So fahamkan how the membrane potential is maintained, is generated, and what causes resting potential to be maintained kan. And then our next lecture we are going to look at the different types of potential that can be generated along the neuron. Okay, so do you have any questions so far? Okay, so if there's no question, I guess we can stop our session for today. So we are going to continue on Friday. Okay, class? Thank you.
Thank you, madam.
Thank you,
Thank you,
madam.
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 Okay. Okay.